My exposure to this entire area of biology began as a medical student in my first year at Harvard Medical School. And George Pilati's discoveries were really fresh, and they were presented in my histology course. And when I saw those electron micrographs of vesicles and endoplasmic reticulum and Golgi and so on, I immediately started thinking, given my background in physical sciences, how in the world could such a vesicle form? What would make that happen? How would it know where to fuse? And I must say, somehow I identified that problem as one that I wanted to solve at some point. And I got fixated on it. And I can't really tell you why, but I just got fixated. I found it as fascinating then as I do now. And so what we did was we wanted to reconstitute transport from the ER to the Golgi. So we took the cells living. We gave them a very short pulse label with radioactive methionine. And, tricks I'd learned in Harvey Lotus's lab, then homogenized the cells to retain them in the G protein in the endoplasmic reticulum. And then we incubated that extract to see if the glycoprotein could be further processed by adding certain sugar residues, especially N-acetylglucosamine, which we knew from the work of Phil Robbins and Stuart Kornfeld and others occurred in the Golgi. So if we could see that processing in vitro, it would have suggested that transport occurred. And indeed, after, a month or two, we observed that was the case. We could see it. But that we had a huge background. Even without any kind of an incubation of longer than a few seconds, even without adding ATP or cytosol, we still saw processing. And what we came to realize was that we already had too much of the G protein sitting in the Golgi, and it was getting glycosylated directly there. So that gave me the idea of what is now known as a complementation assay, which is pretty much the standard design for in those days, most cell-free reconstitutions of transport. And what the idea is, you want to mix two extracts, one from a cell that contains the cargo protein, VSPG, but lacks the processing enzyme. The other extract from a cell that has the processing enzyme but lacks the cargo protein. And so when the two are mixed, if the cargo protein is getting glycosylated, it must have gone from a donor membrane in the extract of the one cell to the processing enzyme in the Golgi of the other cell. And so that eliminated the background that I referred to, because the extract with the VSPG protein was incapable of processing by adding sugar. So when we set that assay up, it actually worked. It was in, I believe, 1979. I still have the gel. It shows a very, very faint band of the VSPG protein being processed to what's called an endo-H resistant form. It was very, very exciting.